So now that you have your data for that lab, we're actually going to be working on the in-class activity for today. And by in-class, I mean just that. Okay? You should be able to finish this or be so incredibly close to done that you require less than 10 minutes of work outside of class to do this. Okay? This is not meant to be more homework for you. Is everybody clear on that? Okay. All right. So what you're doing today is you are simply designing a lab to test the effect of some factor that affects plant growth, because that's what we went over on Friday, okay, on the growth of a plant. All right. So you are only going to have to do the problem, design, hypothesis, and a procedure. Okay, that's it. You don't have to do any analysis, no conclusion, okay, no data, because you're not actually running the experiment, okay, no sources of error. So basically only the first half of a lab report is getting done here. Okay, now, what you're picking here, okay, is the effect of a variable of your choice on the growth of plants. So the point of this assignment is twofold. Review the factors that affect plant growth. Okay, you can pick anything you want. I've had people do things like different watering it with different things. One person watered one with distilled water, one with salt water, one with Gatorade, one with Coca-Cola. Okay, like you can pick whatever you want. Okay, as a manipulated variable, something that is going to affect the growth of a plant. You're going to change it and see what the effects are. Okay, I've had people say, well, I would grow one in zero gravity and one on Earth and see what the difference was. There's no limits to what you can do because you're not actually going to run the experiment. Okay, so you could say I want someone to take one of my pothos plants up to the International Space Station and test it up there. Okay, because
how unrealistic can I get?
Well, I mean, within the bounds of reality. But, other than that.
Realistically, could I test the effect of light on plant growth by putting one 13 centimeters away from the sun?
I'm pretty sure we know the results of that, being as it's a vacuum and really hot. I'm sure that plant will die. Okay, so conditions that obviously a plant can face. Okay, and that can be, again, anything you want. Let's see different colors of light, different intensities, different soils, you pick. Okay, it doesn't matter what you pick. Okay, the second point of this lab is to review the scientific method to make sure you know how to design a controlled experiment. Okay, so I'm going to quickly review what that means. Right? That means you are testing only one thing on the growth of a plant. Okay, so for example, if I was testing the effect of water on plant growth, okay, I would have my manipulated variable be the amount of water. One plant would get nothing. That would be my control group. It will die. Okay, and I'll have one plant that'll get 10 milliliters per day, one plant that'll get 50 milliliters per day, one plant that'll get 100 milliliters per day, one plant that'll get 250 milliliters per day. That one will probably also die. Okay, but all that I'm going to do is test the different amounts of water. Everything else I control. I keep the same. So that means every other factor that we're aware of that control or that can affect plant growth has to be controlled. So I need to make sure they all got the same soil, same amount of soil, same temperature, same lighting, all of those other factors that affect plant growth. You see how this is testing two things? Scientific method, but also your knowledge of factors that affect plant growth. You've got to know what else to control here. Okay? Everyone following? Okay, so you're going to have a problem. What is the effect of blank on plant growth? Whatever it is you're choosing. Okay? Then you're going to have your design, manipulated variable. Okay, whatever it is you're testing, and tell me how you're manipulating it. In that one I just designed, it would be the amount of water received by the plant. Okay, responding variable, growth. That's going to be a little weird because everyone might do that differently depending on what plant they choose. Incidentally, that might be a controlled variable. You probably don't want to compare a bean and a giant sequoia and a carrot in that they are very different. Okay, so what kind of plant are you using? How are you going to measure that growth? Maybe you're going to measure that growth in a number of different ways. Okay? Maybe you're going to measure it qualitatively and take pictures every day. Okay? Or maybe you're going to also measure the height of it or count the number of leaves. You can pick whatever your measures of plant growth are going to be, and I would suggest that it's measures plural, okay, as opposed to just one. All right? So you can decide what you're going to use as your measure and put that in the responding variable. Growth of the plant as measured by this. Okay? Then you've got your controlled variables. You need 3 listed there. Okay? How you're going to control them and why you're going to control them. That is, what effect would they have if you didn't? Same as any other lab. Okay? Then you'll have your hypothesis. If blank affects the growth of a plant and blah, 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 then this is what I'm going to see. Simple hypothesis, same as we've done all along. Then you'll have a list of materials. Okay, that's going to be pretty straightforward. Tell me how big a pot you're going to use, what plant you're going to use, how much dirt. Okay, water is obviously going to be necessary. What kind of dirt? Maybe it's the dirt you're comparing. Okay, whatever it is. Okay, have your list of materials, and then your procedure. Now, in your procedure, that's where I'm going to look to see if you actually control your controlled variables. So when you're describing to me how you're going to set up the arrangement, I'm going to check, okay, they said they were controlling this. Did they control it? They did, okay, good. Right, that's where I'm going to check to see if those controls actually work out. In your procedure, you should also tell them how to record and what to record for their observations. And that's where the experiment ends. You're not actually running this experiment, so that's where it will stop. Okay, everybody follow me there? So, problem, design, hypothesis, materials, procedure. Okay, that's all I'm looking for. And you guys have done that a whole bunch of times already, so it shouldn't take you too long, especially since you get to pick whatever factor it is you're going to test. Okay? All right, go.